So Geet and I are very excited to be here today. And so while Geet gets the demo up and running, I'll give you a little background on what we'll be looking at today. So first, we're playing roles up here. So I'm playing the part of a scientist. You can tell by my awesome science jacket. And Geet will be playing the role of a bioinformatician. So the idea here is that we've taken a set of public TCGA data and we've preloaded it into the platform. So this data is multi-omic in nature. It has somatic variant information inside of it. It has transcriptome information, has methylation information, and copy number information as well. Unfortunately, the public TCGA data did not have associated with it the full germline information. So we made it up. So we put a whole bunch of, synthesized a whole bunch of exome data and put that into our database as well. And so the scenario that I'm going to set up is that Geet and I were standing together at lunch one day, and we were talking about a new Cell paper that was analyzing a line of breast cancer data. And so when I was talking to Geet, I realized that internal to our organization, we had a similar data set, and I wanted to explore that data and see if we could recapitulate a number of the findings that were made in the paper itself. And so after lunch, I returned back to my desk, and if we can switch the screens over to the demo computer, I logged into my DNAnexus platform and was presented with a list of projects that I have available to me. And the only one that I see here is my breast cancer data project. And so I click on that, and I'm brought into this project. Now, most of you all are probably familiar with the concept of projects on the platform, but for those of you who are not, the project here is the sort of main unit of work on the platform. This is where I'm going to store all of my data, whether it be a FASTQ file or a BCL file off an instrument, to VCF files, to, in this case, a large database of information. It's also the unit of sharing. So in this particular project, I can come up here and say, you know who else would really be interested in seeing this data set is my colleague Yichi. I'd enter in Yichi's username, share it with her, and now Yichi would have access to the same data. So for those of you who are familiar with the platform, most of this looks very familiar. But there may be one object that looks a little bit less familiar to you, and that's this guy with the little icon of stacked disks. This is a new object that's on the platform that represents a database object. This is a Spark database, so it's backed by a number of Parquet files that live inside of this project. And if I right-click on this object, you can see that it acts just like almost any other object on the DNAnexus platform. So it has name, and then it has a unique identifier associated with it. And now, instead of a file or a record or other object on the platform, it's this database object. Behind the scenes, again, it's pointing to this set of Parquet files, but we manage all of that for you. Now, I'm a bench scientist. You can tell from the coat. So I'm not that familiar with how to do SQL queries against this database object, but I still want to be able to use the information that the database contains. So we've tried to make that accessible through the use of explorers. So if I click on this database object, I can then click on View and Explorer, and if I keep on hard clicking on that, that won't come up. So what happens now is this is going to perform some SQL queries on my behalf to that SQL data or the Spark database, and sort of start painting up a number of visualizations of what is inside of the data. So I've worked with my colleagues beforehand to set up a default dashboard for the data that is in this database. So what I've asked to be placed on my dashboard is basic sample information on what is contained within this database. So in this case, I have a nice histogram showing the ethnic background of folks inside of my database, an age distribution chart, information about the subtypes of breast cancer that are in my database. In particular, we have about 75% of these come from invasive ductal carcinoma, and about 20% come invasive luminal carcinoma, and then various hormone receptor status. This dashboard is configurable from the end user, so you could select different data to expose in your dashboard. But for this demo purpose, this is what I want to see by default when I first open up the database. I can immediately start doing additional filtering on the data. So let's say I only want to look at the invasive luminal carcinoma. I could come into the Filters tab, go down into the subtype field. And when I click on this, it's going to query the database and ask what are valid values inside of the subtype column of my database. And it's going to pre-populate this list here. I'm going to select invasive luminal carcinoma. And then I'm going to click on Run. So when I click on Run, this is going to query the database on my behalf again, gather the data back, filtering only on those samples that are invasive luminal carcinoma, and then redraw the diagrams that we had seen previously. You can see that it's changed a decent amount. So the age distribution has gotten maybe shifted down a little bit. The hormone receptor status has gone to almost completely ER and PR positive, which is a known sort of hallmark of the invasive luminal carcinoma, and majority of samples are HER2 negative. That's one dashboard that they made available, but you can make as many dashboards associated with a database as you want. So if I click on the 3 dots up here, I can select an additional database. So maybe I worked with my colleagues to set up a second dashboard that compares the ILC and IDC subtypes of cancer more directly, so I can see them side by side and get a little bit more insight into what the differences are between these two different subtypes. So again, this is going to query the database, and it's pre-filtered. Well, I've got the filter on both just ILC at this point. Let me add an IDC as well. So IDC, say Run. And now what we'll see is we'll see side by side the distributions of age for IDC and ILC. And you can see that, again, the ILCs are maybe shifted down a little bit. But you can gain further insight into this. But at this point, maybe I've gotten a few ideas, but what I'm interested in, I don't have in a dashboard yet. I want someone who's a little bit more computationally savvy to come in and help me out with the data. So I could call in my colleague Geet, who's a master bioinformatician, and he can log into a Jupyter notebook and start doing interactive SQL queries directly to the database, maybe to find out a little bit more information about the somatic mutations that are driving these cancer types. So Geet, could you please perform your analysis?
Oh yeah, thanks Brett. So what you see here is a Jupyter Lab interface. Jupyter Lab, for those of you who are not as familiar with it, is like a wet lab notebook for computational experiments. So the main difference from a wet lab notebook, which describes exactly what you're doing and how you did it, is that in computational experiments, how you did it is often described in code. So here in Jupyter Lab, you have cells of code that you can actually execute and see the output of. And you can share these analyses with your colleagues pretty easily. So I could probably go on for the rest of the day about all the cool features in Jupyter notebooks, but what I'd like to focus on is how you can actually integrate Jupyter notebooks with our DNAnexus project. As Fan hinted at, projects are a very core building block in our platform. They contain your applications, all of the different executables for your workflows, the executions, and the provenance of the data that those executions operated on. And all of this can be shared with a colleague just as easily as you would share a tweet, for example. So it's important for us to make available these notebooks in a project-like environment. And that's exactly what you see to the left-hand side here. Just like Brett showed you the interface from earlier where you have the list of files and databases, in the project you see the same thing here where you can see 2 notebooks. There's a lymphoma notebook that currently looks like it's being used by someone else. And then you have this breast cancer database here that is this exact same database that Brett was showing you earlier. So now I'll dig into the notebook a little bit more. The one thing I want to mention here is all of the code examples I'm going to show you are in Python. But you're not restricted to use Python. From this launcher, you can also choose an R kernel, as Fan hinted at in her slides. And you can think of, you can see that there will be analogous code for everything I write in R as well. So the first thing you can do is filter, or you can first import some libraries that are going to be useful to you. So here we have a few plotting libraries and data analysis libraries. And the next thing you want to do after you've imported some libraries of interest is, aside from reading data from files, on our platform, we have the ability to connect to Spark databases, and you can bring that data into your notebook and start to analyze it. So I've hard-coded a couple of things to connect for this demo, but what it comes down to is a one-line connection function. Once you connect to that, you can basically run any database query you want and bring that data into your notebook for analysis. So what I did here was I just did some simple show tables from query, which just lists all the tables in the database. And you can see here there's a variety of data available to us for this breast cancer data set. I dug into it a little bit more and started looking at the mutations table. So this is just selecting all rows from the mutation table. And you can see here there's a lot of different information available to us for the mutations. As I started digging into it a little bit more, I see this consequence column, which contains different kinds of variants and how they could affect potentially the function of the resulting protein in that gene. So what I did was, for this data set, I want to look at mutations that might affect the function more drastically than others. So I came up with a list of about 5 of them. And I wanted to create from that a simplified table that just said, for every row in this table will be a mutation. I just want to keep the gene it's associated with, what sample, and whether it's a loss of function for my definition of a loss of function. So I continued to use that table to just make a simple plot of the genes that have the most loss of function mutations for that definition. And you can see here it's really easy to visualize in just a few lines of of code in Jupyter. But there's some odd things, like there's this gene TTN here, which is a very long gene. And while this is interesting at first glance, this very long gene, for example, may have a lot of loss of function mutations just by chance. Or another way to look at it is you may want to compare the number of loss of function mutations in a gene to the number of non-loss of function genes. So to that end, I just created a quick scatter plot where each dot in the scatter plot is a gene. On the y-axis, you have the number of loss of function mutations, and on the x-axis, you have the number of non-loss of function mutations. And the idea here is that by chance, you would expect to see maybe a similar number of non-loss of function mutations to loss of function mutations. But for genes associated with this breast cancer data set, maybe we're interested in genes that have way more loss of function mutations than non-loss of functions. So as an exploratory data analysis plot, you can see that that would show up as outliers here. And we do see a few outliers. So once I verified that there seem to be some of these outliers, I wanted to get a list of what those genes actually were. And so just with a few more lines of extra code, you have this table that looks at the ratio of the number of non-loss of function genes, or loss of function genes to the number of non-loss of function genes. And you can see here that the TTN gene that did show up before no longer is in that list. So I'm pretty happy with this. Brett just gave it to me. I did an initial analysis, and I'm getting a little tired now, and I know Brett's a night owl. And I just thought I would send this his way and see if he can see what I did.
So, in general, what I'm doing is I'm examining through the notebook that Geet put together for me, and it looks fairly good. The nice thing is that I, as a scientist, don't have to code. I can just look at the work that Geet has done himself and look at and reuse his code that he's put together. One of the things that I found when I walked through the data is that Geet may have misclassified one of the mutation types. So in his list of types of mutations that were loss of function, he claimed that a missense mutation was a loss of function. And I say, oh, well, that's not right, Geet. I can easily comment out that line. Even me, the computer novice, can do that. And rerun his exact same notebook and see what of the results sort of still hold. Once I've gone through the notebook, though, maybe I have further analyses that I want to perform. Maybe that spurred additional hypotheses on my part that I want to go and explore. So what I can do then is go back to my project. I can again, view and explore the database in question. And maybe at this point, I decide, you know what, that's loss of function information was really interesting, and I'd like to set up an additional dashboard to help me to view that data without having to go into a notebook. So I could collaborate with my users, and maybe I want to kick off an association test that's comparing IDC versus ILC. Is look to see if there's any germline variants that are indicative as to which type of subtype of breast cancer the user is going to create or develop. And so here, what I'm doing is I'm going to create a filter so that I filter just on the subtype of IDC cancer. So I'd click on that, select IDC, I believe, and then run this as my filter. And what we want to do is make it super easy for you to save this filtering into an object that we call a cohort. And so once I've performed this filter, the dashboard is going to be updated as we've seen before, and then I will be able to click on the Save as Cohort object, or link up at the top, and save this as a cohort of IDC. This object on the platform is just a simple small record saying what my filter criteria is. So it's very lightweight, and I can share it with whomever I want to and create as many cohorts as I want. So next, after I've created my IDC cohort, I will come back here and change to an ILC cohort and save that as an object as well. So that will take a few seconds. So click ILC, run that. That will actually perform the SQL query against the database itself. The dashboard will once again be updated, and then I can save this as a new cohort. That will be ILC. So now at this point, I have two cohorts on the platform. And maybe I want to do a case control association test comparing these two cohort objects. And I'm going to click on the Start Analysis button, which will bring up a list of apps that will be able to operate on these cohort objects. So it's going to populate a list of apps. I'm going to select the GWAS app because I want to run a GWAS on these two cohorts. I'll click on that and say Start or Run Analysis. And that's going to come up with the new workflow executor that Fan demonstrated earlier, pre-populated with my IDC cohort. I will then click on the Select cohort and choose the, it's pre-populated with ILC. I'll now add the IDC one. And now I specify where I want to store the results, a new database object. I'll put that in, and then I can click on the Run as Analysis. And what will happen is it'll launch a new set of jobs to actually perform the GWAS analysis itself. Now that will take about 20 to 25 minutes to run. So I've short-circuited it to a previous run of the GWAS itself. So let's look at what that looks like real quickly. So this is the results of a previous run. And it brings up a Manhattan plot showing you the p-values for the various genomic loci for IDC or ILC subtypes of cancer. So anyway, that gives you a brief overview of what the translational platform looks like and the key features that we wanted to highlight and how the different user groups we envision might be able to use it. So anyway, I thank you for your time, and I'll